Hello everyone, this is Dr. Sachinandan Datta, and in this marine ecology series, today I'll talk about the sampling strategies and biodiversity measurement. So first we see what is sample and why we do need to sampling. Okay, so sample is basically a part of the population, or it is called subset of the population. Basically what happens, you cannot practically count the every single organism of a population. You cannot count all the organism in a single population. So that's why you need a sample of the population. So sampling provide a representation of the number and the distribution of an organism. So sampling is provided your representation of the whole population, and there must be a sufficient number of samples. So you need to estimate a sufficient number of sampling from here, everywhere, and it should be basically represent the whole situation of the population. So basically the sampling is the subset of the population. So what are the sampling methods or sampling techniques? So there is basically two types of sampling methods or technique. These are the probabilistic or the random technique and the non-probabilistic technique, and that is also called the non-random technique. So in probabilistic technique there are different types. These are the simple random sampling. So what is simple random sampling? Simple random sampling means in a population you do sampling from here, there, anywhere. So it's the random sampling, simple random sampling. You just collecting your sample from anywhere. Then the systematic sampling. Systematic sampling means you are collecting the sample from in a particular area and systematically. Okay, and also you are collecting sample in a particular time period, or particular time period of a sampling time is fixed. So that is a basically systematic sampling. Then the stratified sampling. Stratified sampling means you are collecting sample from the strata of different strata, just like you are collecting sample of the different age group of the population. Just you are collecting sample from the young age group, then the adult age group, or the mature age group. So this is basically strata of different strata of a population. So that is a stratified sampling. Then the cluster sampling. Cluster sampling means you are collecting sample from a cluster of the population. You are collecting from here is one cluster, three cluster, and two cluster. So you are collecting sample from a cluster of the population. Then the area sampling means you are collecting sample of a particular area, only the particular area of the sampling. Okay, particular area of the population. So that's the area sampling. Then the multi-stage sampling. Multi-stage sampling means you are doing sampling from the different stage. Just a simple example, you are collecting sample from the whole state, then you are collecting sample from the district wise, you are collecting sample of the village wise, you are collecting sample from the household wise. So it's a multi-stage, so different stage. So 1, 2, 3, 4, 4 stage. So that is called a multi-stage sampling. In basically this sampling technique are used in scientifically we are using much more of this kind of sampling techniques. But in case of a non-probabilistic or non-random sampling technique, these are basically used in social science. These are basically questionnaires. So lots of questions are set, and then you do sampling from the questionnaire. So it's a judgment sampling, then the convenience sampling, then the quota sampling, then the panel sampling, then the snowball sampling. Just one example I'm telling. The snowball sampling. Snowball sampling means you are asking the questions from one individual, then this individual is asking the questions of another individual, and he is also asking to another individual. He is also asking another questions to another individual. He is also asking from another. So it's then called a snowball. So they are increasing. So this is a snowball sampling technique. So this much more a questionnaire, and basically social science people are using much more this kind of sampling technique. And now comes to the different sampling technique in biological diversity measurement. Basically we are using this sampling techniques in any kind of density or abundance study, or the population density study. So these are the basically a few techniques are there. First technique that, and this is most simplest technique, that is called quadrat sampling. Quadrat sampling is nothing but a square box you are preparing. It could be metal, or it could be any material, or it could be wood. Any material, you just make a square, and that square is according to measurement, it could be different. It could be 1 meter square, it could be 2 meter square, 5 meter square, 10 meter square. So whatever you need, you just prepare your square, and you plot this square in your study area, and it should be random location you are plotting your square, and the counting the individual inside the square, and then counting the plant or animal, whatever you want to estimate, and inside the your square, and you always ignoring what is the outside of the quadrat. Okay, so basically this square is called quadrat, and repeat this process. You have to repeat this process to much more accurate of your result, to get accurate result. Okay, and then calculate the average number of individual per square unit square, means how many number of individual average unit square for each species, and then measure the area of habitat under the study. That estimate basically the density of the species. You can easily estimate it from the quadrat sample. So how do you get the density? So density is very simple equation. For this equation, you have to know the total number of individual of a species in all quadrat. So suppose in you are do 10 quadrat sampling, okay, and total number of individual of all species in all quadrat. So you are doing 10 quadrat, and total number maybe the 100. Okay, and so that is the total number of individual of a species in all quadrat. So 10 into 10, so 10 individual and per quadrat, and 10 quadrat, so 100 individual in all quadrat. Okay, and then by total number of quadrat, 10 quadrat, and area could be 1 meter square, 2 meter square, 5 meter square, whatever your measurement. And that technique is used everywhere. It could be used in the forest, it could be used in marine sciences, it's used in the beach area, in sandy beach or rocky beach, and also in the crowded ecosystem, or the bottom sea shore, or the sea bottoms, means sea bottom. You can also estimate the different density of the different species. So that's a very simplest technique to measure the density or abundance of the species. Then the transect sampling. Transect basically it's a line. It's nothing but a sampling line that covering the interest of your interest area. Okay, and in this line you want to find your sampling plots. Okay, so transect means this is your total habitat area, and you just plot a line on this habitat area, and you just start sampling from this point, and following this line, and you when you are going to through this line, and you just counting your whatever organism you are getting your observation, counting this organism, and that distance also, that distance you have to measure. So this is a transect method. So the transect is basically two types. One is the line transect method and belt transect method. So this is basically line transect method, and this is basically belt transect method. We will see later on. So line transect basically single line and straight line throughout your habitat area. Okay, and belt transect it's a type. It is basically a composition of the several quadrat, and it each and every quadrat you are means gathering all the quadrat, and you are making in a line, following a line. You just do quadrat from in the both side of the line. So that's a 1 meter wide. You can measure any type. Okay, so that's a belt transect. And this sampling or the transect sampling could be continuous. If your habitat is small, then you make a continuous, or if your habitat is very large, then you can make a interrupted. So you are measuring from here. So one transect, another transect, and one transect. So this is a interrupted sampling. Okay, and continuous if your habitat is small. Okay. Now the line transect. So line transect is basically you fit a rope or line inside your habitat. Just hold the habitat, you fit a rope, and in following the rope you are just counting your organism, whatever you are getting means you are observing. So this is like a rocky shore region. You just plot a line or a rope, and according to rope you are whatever organism you are find in your path, so just measure this one. Okay, And on the way, so this is observation, you are observer, this is your line, you are going, and your object is here, so you have to measure the distance from you to the your object, and also you measure the angle, and you also get this from line to the distance of this direct distance of this. So from this calculation, you can also calculate the abundance of the species or different individual of that habitat area. So line can be chosen, you can choose the line randomly, or it can be choose from the environmental gradient, or it could be choose as a slope. So these are the different technique why you are choosing the which line. Okay, so density you can easily estimate from this line transect. So D is equal to N by 2LA. So D is the density of the organism, N is the total number of the animals seen or in your transect, and L is the total length of the transect, from here to here. Okay, total length of the transect, and A is equal to basically half of the effective stripe, also half of the effective stripe. So A constant that could be easily estimated. So from this equation you can also estimate the density of the species. Then the build transect method. Build transect method is nothing but a group of small quadrats. So join together, you just bringing all the group of small quadrat, and you just making a build transect. So build transect method is similar to the line transect method, but it gives you more information about the abundance and also the presence and absence of the species, because you are making according to line, you are making a quadrat, or through the line you are making quadrat of several number of quadrats. So here your probability of getting the species is much more, and so abundance is very accurately you can estimate the abundance, and also you can estimate the absence of presence of any species in your belt total area of the belt transect. Okay, and it is basically used in different ecosystem. You can use it in the forest ecosystem or any grassland ecosystem, and in marine ecosystem it is basically used in the sandy shore area or rocky shore area, or it can be used in the coral reef ecosystem also. So from the belt transect, so what are the number of quadrat? Basically the number of quadrats already sampled in the area. What are the number of quadrat you are sampling? Almost 1 2 3 4 5 6 7, there are 15 already quadrat are there, you sampled 7. Okay, and is equal to total area of the unit. So whole area means that is the total area of your measurement into total number of quadrat to be sample. What you want to do sample again. Okay, again means next, and by the total area of the habitat. It's the total area of the habitat. So this is a very simple equation. You can also estimate the number of quadrat in the unit. Then the Mark-recapture method, it's a very interesting technique. So it's basically estimate the abundance also, and also you can estimate from this Mark-recapture technique. So, and this technique is what happen in this technique, you just capture any animal and then mark it and then release it, and then again you captured it and you check whether it is marked or not. If it is not marked, again means unmarked animal you find, then again marked it. So this is a technique that you animal you captured, then you count their number and then tag it and then release it, then again captured and check whether it is tagged or not, or whether it is marked or not. If not, then again marked it. Okay, and so that is a technique of mark, capture and recapture. Okay, so that's a technique very much effective to estimate the abundance. It is very accurate. So it's a very simple equation. You can also estimate the abundance from the mark recapture. So it's a n1 into n2 by n3. So what is n1? N1 is the total marked after catch 1. So after capture 1, total marked of the organism. Then the n2, what is n2? Total marked after catch 2. After catch 2, whatever the number you are giving, again mark. And n3 is the total caught of in catch 2, total caught in the catch 2 that are marked in catch 1, that already marked in the catch 1. So this is the n1, n2 and n3, and by putting the simple equation you can calculate the abundance from the mark recapture methods, and it is also very commonly it is called the Lincoln index. Then I have already talked about the biotic component, how you can measure the biodiversity. Okay, now along with the biodiversity, now some biophysical or the physical factors or the abiotic factor need to be measured. So this is also means after doing the biological measurement, if you do the abiotic factor measurement, the environmental parameter, that could gives you a total complete study or complete knowledge about the whole ecosystem. So that abiotic measurement is also very important along with the biological measurement or biodiversity measurement. So what are the abiotic factors? So there are lots of the abiotic factor: temperature, salinity, humidity, then the DO, then the different nutrients. So different techniques are there. So what are the instrument we are using? We are using the light intensity, maybe measured by the light meter, and it could be measure the intensity of the light, and it is also called that light intensity. You can also measure the PAR, photosynthetic active radiation. Okay, then the temperature, simple digital thermometer you can use to measure the temperature of the water surface or maybe the air, whatever you want. And the air movement, the anemometer, it's basically used to the air speed or the wind speed, and also you can get the direction. Okay, and dissolved oxygen, it's a very important parameter for the physical parameter among the abiotic factor, that is measured in diometer. And then humidity, hygrometer, then the salinity is easily measured by the refractometer, and the pH is very important parameter, it can be used in the pH meter. And any different other organism techniques to measure the current meter, then the nutrient you can measure all the nutrients. So these are the lots of the abiotic factor also need to be measured along with the biotic factor. So this is the biodiversity measurement, different means indices. This slide I have already talked in my biodiversity lecture, but here I also just little bit discuss, and we will also discuss all the technique of the biodiversity measurement technique as a practical in a Excel sheet. How we can calculate all the with example we will see in the practical session in Excel sheet. So here by diversity measurement, you do that all the diversity indices. So most of the important 4 diversity indices, there is also more diversity indices, but most important is the Shannon-Wiener, Simpson, and the Mark-recapture richness, and the Pelu's evenness. Okay, so Simpson diversity index and Shannon-Wiener diversity index. So this is the very two important diversity index. So Shannon is very important. That's H bar is equal to that. I have already discussed in my biodiversity lecture. You just go through it. So it's Ni by N into ln of Ni by N. Ni means the total number of individual in of species of a particular species of X species, and N is the total number of all individual. Okay, and diversity index, Simpson diversity index, D is equal to nothing, 1 minus sum of N minus N. N is the number of individual of a particular species, N is the total number of individual of the all species. Okay, Mark-recapture richness index. So how your means habitat is rich in species diversity. Okay, so richness index is very important. So richness is nothing but S minus 1. S is the total number of the species minus 1 by ln of N. N is the total number of individual in the sample. Okay. And evenness index basically calculated from the Shannon-Wiener index. So evenness is equal to Shannon-Wiener index by ln of S. S is the total number of species in the sample. So these are the 4 very important biodiversity measurement. These are the called the diversity indices. So we'll check in practical session. So species richness. So species richness is basically it's a qualitative description of the habitat or qualitative description of the ecosystem. You can or whatever your study area. So how many different species can I see? If you see much more, so this is rich means you are seeing that how many 10 species. Okay, 10 species. So this is rich, but this is not actually very rich. When it will be rich, each and every species, the number of the individuals, suppose it is 10, and this is 5, 2, 1, 1. So this is not that much rich, because each and every species must have their individual number will be very high. Then if you check this one, 9888, and 9 or 7. So this is much more rich. Okay, this is much more rich because each and every species individual number is also high. So that is the thing, that more species does not always means the more biodiversity. Why? Because there may not be the many individual, like number of the individual or number of the means individual of the particular species should be high, then it is called a richness. So that number of the individual of that of the species, that is also called evenness. So evenness, what is evenness? It's basically quantitative. It's an quantitative measurement. You are measuring the number of individual of each species. So it's then a measure of abundance of each species. And Pelu's evenness index, it's basically, I have already talked, Shannon-Wiener by a log of S. And if your Shannon-Wiener index is equal to log of S, mean total number of species, or log of total number of species by Shannon-Wiener, if it is, this is equal to, so that means your Pelu's evenness index is 1. Okay, so when you get close to 1, means it's a very highly even. Okay, evenness index is very high. And if Pelu's evenness index close to 0, means it's an uneven index. Okay, that simple example we'll see here in two sites, one site and site 2. So in site 1 there will be 4 species we found in both the site. In site 1 you can find the 12 individual, 11 individual, 14 and 13 of the 4 species. And so, and site 2 you got a species A 49 and other are very less in number. So from this you can easily told that you can easily tell that site 1 is much more even, because here evenness index is almost 1, so it's 1. And in site 2 evenness index is almost very low, only 0.2. If you calculate, you can calculate, so calculation is here. So that's easily from this data you can also estimate. Okay, and so this is much more even and this is very less even. Okay, and abundance can be measured in percentage cover. So how many percentage of each species occupied of that particular of species, means of that particular region or particular area. And the population density I have already talked about it. In total area, how many number of individual are there, so individual per quadrat. Okay, and the species frequency means if you from this quadrat or quadrat 1 2 3, if you want to check that species A, how many times are you getting species A, so this is the frequency. If you get this in all the quadrat of species A, means the frequency of species A in all the quadrat is, frequency of the species A is high. And so this is the total statement, that if your species richness and species evenness is very high or increased, then the biodiversity of that particular region is also increased. So richness is increased, means number of species is increased, and evenness means number of individual of each species is also high. So then your biodiversity is also very high. So there is a very simple means table from you can also calculate that, check and see that which field or which area is much more biological diversity high, because here you can check that the individual same species, so richness is same species, richness is same, but evenness is very low here. Okay, and very high here. Okay, so species site B or field B is much more diverse. Okay, now Simpson diversity index. So if your diversity or D is very good, means your habitat is much more diverse. Diversity index is very high, means your habitat or your region is ecosystem is much more diverse. Okay, and species richness is also very high, and it also, you can also tell that the environmental impact, any kind of environmental impact it can tolerate, because here the species richness is high. But if your D is very low, or species diversity index is very low, or Simpson D is very low, and then there are low number of species, the richness is very low also, and if there is any kind of change in the little change in the environment, or like pollution or the temperature, that could be hamper the whole ecosystem. Okay, so that is this is the interpretation of your result. And one of the explanation of the result that if D is almost 0.56 and D is in field B 0.7, so D is very high in this field B. So from this index you can also tell the same thing I have already explained, that in field B is much more diverse, much more rich, much more evenness is very high. And if there is any little bit changes in the field A, it harmful to the whole ecosystem. Okay, so there the area is more likely withstand changes in the such as the pollution or temperature, any kind of changes. Okay, it's much more harmful for the field A instead of field B. Okay, so that's all about the species diversity indices or the biodiversity measurement and techniques and the measurement. Okay, so thank you. If you have any query, just comment me. Thank you.